Good morning, guests. Welcome to our long and short read sequencing workshop. My name is Esther, and on behalf of Oxford Global, I would like to thank you all for joining us today. We have three amazing presenters joining us today. Patrick Descombes, Head of Genomics at Nestle Research, who will be providing a general introduction on long-read and short-read sequencing technologies. Patrick will be followed by Professor Massimo Delledone, University of Verona, on characterization of long DNA regions by short and long reads, and Dr. Edward Oakeley, Associate Director of Genomics at Novartis, on targeting long-read sequencing. The presentations will last about 50 minutes, and after all our speakers deliver their talk, we are going to have a Q&A session. Please submit your questions through the chat feature of the platform or raise your hand to ask your question directly from our speakers. And now, over to you, Patrick.
So good morning, everybody. Thank you, Esther. Can you hear me?
Hi, Patrick.
Yes, we can hear
you.
Fantastic. Thank you. So good morning, everybody. My great pleasure to participate to this webinar. First, I would like indeed to thank very much Oxford Global for the organization and thank my two friends, Massimo and Edward, who kindly accepted my invitation to participate to this webinar. So the workshop was presented by Esther. Thank you. So I will skip that one and directly go into the meat with my brief introduction. As you know, today we're really in the middle of a fantastic revolution of genomics technologies with an amazing increase in the throughput and the reduction in the cost of these technologies, which allow to deploy them in different fields and different research themes, both for human genetics, but also for plants and bacteria, just to cite a few. Along with this development, we are generating more and more data, enabling the so-called large-scale studies and contributing to the big data, which go along with the need to store, process, and analyze this data, and of course, the concomitant development of bioinformatics and biostatistics. We also know that these technologies are evolving very fast and becoming more and more easy and standardized. Still, they remain quite sophisticated and complex and require dedicated personnel to be wisely and well used. Now, the impact of genomics is huge, and I have here cited a few examples which are by no means exhaustive, just to show how the imprinting of genomics into the daily life is amazing, not only from the initial knowledge building that we all have been involved in, but also moving towards such examples such as personalized medicine and amazing progress in the treatment of some diseases like cancer, or improvement of crops and improvement of breeding programs. Not to cite also food safety or microbiome profiling. And I will stop here this list, but as you can see, there is really a major spread of these technologies in our daily life. Now, going into the meat of this workshop, I would like to briefly here summarize some definitions on the difference between the short and long reads. When we talk about short reads, we talk about technologies that generate fragments of 50 to 500 bases, which are mostly used for resequencing purposes or sequencing of species identification or RNA profiling. One of the characteristics of this type of technologies is the amazing throughput and the very high accuracy. On the other hand, long reads technologies are technologies that provide sequences of fragments of thousands, tens of thousands, hundreds of thousands, or even megabases lengths with a slightly lower accuracy. And they're extremely useful and helping for studies such as de novo genome assembly, and in particular, the resolution of complex genomic regions such as repeats, also for phasing haplotypes, and of course, characterization of isoform of transcript. And finally, some kind of in between, we have the so-called synthetic long reads, which are basically the generation of long reads starting from indexed short reads, which again are here extremely useful for de novo genome assembly or characterizing complex regions. Key, of course, is to realize that not any of this technology is ideal, and often we need to combine some of them, but really more importantly, to choose which is the best, depending on the need and the purpose of the studies. And again, sometimes the combination is a wise choice. Now, without going through an extensive list, I wanted to highlight here some of the key players in the short and long read technologies. And of course, to start with short reads, the three main players are Illumina, IonTorrent, now Thermo Fisher, and MGI. Both for Illumina and Ion Torrent, the template which is sequenced is basically a piece of DNA of interest flanked by adapters which allow PCR amplification followed by immobilization either on the glass slide or on the bead. In the case of Illumina, the DNA is amplified on the glass slide in a so-called solid phase kind of PCR, which generate clusters of molecules which are subsequently sequenced by synthesis using a mix of 4 nucleotides that are labeled with different colors depending on the version, or now 2 colors or even a single color depending on the evolution of the Illumina system that you have. In the case of Ion Torrent, the system is using an emulsion where basically one fragment of DNA is attached to a bead, and in an emulsion clonally amplified, this bead is then dropped in a semiconductor device which will basically measure the incorporation of each of the bases sequentially flown onto the system by measuring the release of protons. So basically the Ion Torrent machine is a pH meter, if you wish, and generating from this a sequence of interest. One main difference between Illumina and Ion Torrent is their faculty or ability to deal with similar sequences or amplicons. In the case of Illumina, this is a bit challenging because the system needs different bases to identify clusters at the beginning, whereas for Ion Torrent it's a bit different, and then a single sequence, for example, can be used. There are, of course, other differences that I will not go into details. And finally, more recently, a player in the field is MGI, which combines technology from Complete Genomics and is a spin-off of BGI. And here, interestingly, the generation of the template for sequencing is based on a rolling circle amplification, certainly impacting and reducing the propagation of errors in the amplification process. Interestingly, also, the system to identify the base following the sequencing by synthesis protocol that they have, where they use antibody that recognize modification of the 4 bases, and this allows an improved signal-to-noise ratio. And of course, the community is waiting to see how these different technologies are compared and performing each other. Now, moving to the long-read sequencing technologies, the main player currently are Oxford Nanopore and PacBio, Pacific Biosciences. In the case of PacBio, the template of interest is flanked by a hairpin to which DNA polymerase is attached, basically generating a circular single-stranded DNA molecule that is sequenced in a ZMW, or a small kind of nanopore, on this smart cell. This smart cell currently can hold 1 million wells. And the system basically records the incorporation of DNA by taking movies of the incorporation in the active site of modified nucleotides. And this movie is translated into a base sequence. In the case of Oxford Nanopore, the template is generated by ligating adapters and hooking a motor, an engine that basically targets the molecule to be sequenced on a nanopore and allows the transition of this molecule through the pore. During this transition, the peak, the fluctuation of the current between the two sides of the pore is measured, and it is then converted into a base sequence. The difference between the two technologies is in the length of the reads. For PacBio, we talk about tens to hundreds of thousands of bases in the best cases. For Oxford Nanopore, this can go up to higher read lengths, up to megabases. And the error rate, which is in the range of 10 to 12% for PacBio and in the range of 5–6% for Oxford Nanopore. Notice here, however, that in the case of PacBio, owing to the circular molecule that is sequenced, the same molecule can be sequenced on the forward and the reverse strands multiple times, allowing to generate a circular consensus or high fidelity reads, which has a very reduced error rate. So again, here a complementarity between these two technologies. And finally, let me briefly introduce additional type of application or technologies to complete this introduction. The synthetic long reads, either from 10x Genomics or MGI, which basically allow to isolate individual DNA molecules either in micro droplets or around beads, and then tag them with indexes and allow to sequence them with short read and then assemble them using the indexes together into a single molecule, allowing with the sufficient coverage to again generate high fidelity phased fragments of DNA. Let me just note here that 10x Genomics product has recently been discontinued, and we will see what's the evolution in this domain. Finally, two additional elements that are of interest when we talk about working with combination of short and long reads. First, high-C proximity ligation, which is currently mainly proposed by Dovetail, where here the DNA is cross-linked, followed by enzymatic digestion and ligation to generate chimeras of fragments that are adjacent in the nucleus or in the DNA. And those fragments are then sequenced. The frequency of interactions between the fragments being proportional to the distance that separate them in the DNA, this technology allows to generate proximity or frequency map that can be used then to scaffold contigs generated from another technology and generate chromosome scale assemblies, as you can see here with this example of the assembly of the coffee genome of Canephora that was produced in my lab a couple of years ago. And finally, another very interesting development, which is not really sequencing, but really visualizing DNA, and it's the optical mapping developed by BioNano Genomics, where DNA fragments of very long lengths, up to chromosome lengths, are labeled using a nick produced by a rare cutting restriction enzyme, followed by a gap filling, and then separated on a capillary device on the instrument, an image generating this type of restriction maps that can then be compared with contigs coming from other assembly technologies and allowed to bridge contig together and again here contribute to scaffolding genomes. Now, when we talk about these technologies, there are additional elements which are extremely important. First, the input material, and here we talk about DNA extraction. Indeed, the development of these new long sequencing technologies has forced the community to reevaluate the methods to extract DNA of appropriate lengths, because of course there is no point to try to sequence 50 kilobases if your fragments of DNA are 10 kb after your extraction. So this has opened again a lot of development using different methods to ensure that the yield, the size, and the purity of the DNA is prepared in an appropriate way for the method of choice. And last but not least, the development of this technology, of course, requires a dedicated standardized pipeline for the base calling, the quality evaluation, and the error correction, but also all the downstream applications, should it be to assemble genomes, to identify variants, etc., etc. And we see here a fantastic development and in improving in the standardization of these methods, although we all acknowledge that there is always room for that. So to conclude this very brief introduction, let me summarize by stating here that we see a fantastic development and a good complementarity between short and long read sequencing technologies. We witness a much welcomed increase in the standardization of the protocols and the data analysis, although everybody in the community acknowledge that there is always room to improve this. And I think that we can say we are really in an unprecedented situation with opportunities to use these technologies for discoveries and knowledge building with the application that was highlighted in the initial part of this presentation. And really very important here, the choice of the approach and the technology is really guided by the question that has to be addressed. So with this, I will stop this very brief introduction and hand over to Oxford Global to introduce Massimo for his talk. Thank you.
Thanks very much, Patrick, for this great introduction. And now we will move on to our second presentation today from Professor Massimo Delledone, University of Verona. Over to you, Massimo.
And thank you, Patrick, for the great introduction. So that saved me a lot of energies to explain the technologies. And especially, I have to say that I agree very much with the point that DNA extraction is now becoming the bottleneck of the long read technology. Anyway, the sequencing technology, as we have seen from the introduction that Patrick gave, it's improved very much in the last few years. And I like to say that now sequencing is like fishing. You can use a rod and catch a single gene or a single piece of DNA, or you can use a net and catch the entire genome. Now, the problem is that when you go fishing and you want to go fishing a certain kind of fish, you need to choose the right bait. And the technology that we have now available in order to sequence and to capture a short reads have been based so far on PCR and long PCR and on panels in hybridization capture that were then sequenced with the short read. But now there are new technologies available. Many of you knows about Cas9 mediated capture. Probably not so many of you knows about the very interesting technology that I'm going to describe now, that is the X-drop technology developed by Samplix, which allow to capture very long fragments that can then be sequenced by short and by long reads. This is the technology. In brief, you chop the DNA in very long pieces, maybe even 50, 80 kb. And then you encapsulate every single fragment of DNA in a droplet. Then you monitor the droplets containing your target DNA by simply performing what, in my opinion, is identical to a digital PCR. So you design two primers that allow you not to amplify the entire region because it's too big, but only to amplify a very small region that allow you to know that the positive droplet is containing DNA that is in the region where you designed the primer. And then you sort the droplets with a size sorter, and then you can perform a multiple displacing amplification in order to amplify the DNA. That, of course, is very limited because it's coming from a limited number of positive droplets. And then you can sequence that DNA by using short reads or long reads. Now, we tested this platform on FMR1, a gene that for us is very much interesting because, as you can see, it has a very high GC content. The first exon has 77% of GC. It has single nucleotide variants and indels, which are causing disease. It can have a repeat expansion that can be very long. And there are also possible interruptions at the pre-mutation stage that keep the expansion limited. When the interruption disappears, usually there is the expansion of the repeat and the mutant phenotype. So in a system like this, we decided to test 3 samples. The first sample is a normal sample where we expected to see a small number of repeats, so an expansion of 90 base pair, a mid sample at the pre-mutation stage that was reaching a site that was a little bit longer than the classical Illumina sequencing read that is based on 150 per end. And then a mutation stage in which one of the allele was reaching a site that was probably longer than 600 nucleotides. Then we prepare the DNA for X-drop. As you can see, the starting material is very limited. This technology just requires 10 nanograms of starting material. And then we went through the process and then we sequenced, and you can see the coverage of the region that we were targeting here. You see the coverage both with nanopore and Illumina. The coverage was good. You see that there is an enrichment of about 80 kb that you can control depending on the fragmentation of your DNA. But the coverage that we got, it was quite limited. This technology is working, but in our hands, we still have to optimize it. So what we are going to see are some preliminary results that for us are very much encouraging. But in this case, you see that the coverage that was reached by Illumina was not very high, apart the first case. By Oxford Nanopore, it was quite low. And since our first goal was to call the variants, of course, we did not expect to find a good concordance rate between Illumina and Oxford Nanopore. Illumina can call the variants at very low coverage. This coverage was too low for Oxford Nanopore, so we cannot really compare in this case. But when we moved to expansion analysis, the data that we got, although the coverage was so low, they were very promising because, as you can see, the expansion that has been reported by Illumina and Oxford Nanopore is identical to 28 CGG plus 2 interruptions that are properly positioned both with Illumina and Oxford Nanopore. And when we analyze the sample that was expected to have at least 200 repetitions, we found that Oxford Nanopore, despite the low coverage, was able to detect an expansion of 369 triplet. That is a fragment of more than 1100 nucleotides. And of course, here, Illumina failed. The short reads of Illumina did not allow to reconstruct the entire repetition, of course. So this is a case in which Oxford Nanopore is complementing the Illumina data. And when we analyzed the 3 samples, we found that there was a good correlation, fantastic correlation, of course, when the repetition is small and the Illumina can measure it. But Expansion Hunter, that is the software that we use to measure the expansion with Illumina data, failed to properly reconstruct the expansion when this was longer than the classical 150 nucleotide. You see here of the sequencing. Here it has to be almost 120, and Expansion Hunter found 58, and here instead of 369, Illumina reported only 53. Then we decided to try to go even longer and to test other diseases in which the expansion can be much longer. In this case, the DMPK1 that is causing this myotonic dystrophy type 1. You see that the expansion of the mutation stage can reach 3,000 triplets, that is about 9,000 nucleotides. This is the classical way people look at it by long PCR. Another expansion that is even longer because it can reach 44 kb. 44 kb, like in the case of CNBP, it's an expansion that is impossible to measure by even long PCR. And the technology that people use are like this, Southern blot. So you run a gel and you look at the bands. And as you can see, there are many bands here because in this case, you don't have only an expanded allele, it's a mosaic. So every cell may have a different expansion. So because of that, it becomes very complicated to characterize this long DNA fragment. In this case, we decided to switch to CRISPR-Cas9. The kit is now, I think, available from Oxford Nanopore. The system is very simple and it's very straightforward. As you can see, in less than 2 hours, you can simply remove the phosphate of your DNA, you cut with your guide, you attach the adapter, and you start sequencing. And we tested samples carrying a long expansion for FMR1, and a medium expansion for the DMPK1, and a long expansion for CNBP. We designed the guides, and we found that different guides perform differently. This is a case in which you can see 2 pairs of guides 1 and 2, and guides 3 and 4. And you see that on the same sample, the guides 1 and 2 are able to produce a very high number of fragments that start and end exactly where the guide is cutting. Whereas for the guide 3 and 4, the efficiency was much lower. So you see a lower number of proper fragments. When you see longer fragments, they are due to the single guide cut. So these are fragments that have been cut in one side and not in the other side. Okay, we analyzed these 3 samples. As you can see, the first one is the one that has been already characterized by sampling before. Then there is a DMPK1 with a good expansion reaching 1,800 nucleotides, and an expansion for a CNBP that was more than 10 kb, not really very well characterized. I forgot to say that the good thing of CRISPR-Cas9 is that you can first put together many different guides. So that means that from the same sample, you can capture, in this case, both the region of FMR1 and all the other regions. So you can go multiplexing, not only in terms of guides and so of the target region, but also of samples. In this case, we have been capturing 3 different samples, and each sample were having guides for the 3 genes. So we were characterizing 9 regions in just one single sequencing run. This is the coverage that corresponds to the reads on the target, because remember, in this case, the reads are not distributed along the target region. They are starting on the beginning of the target region, and they are ending at the end of the target region. So this represents the coverage, quite a high coverage. And you can see here an example of the quality of the enrichment of the 3 regions of the samples. That is quite good. And you can see here how it has been possible for a CNBP, for instance, by looking at nucleotide, we were on this fragment to see that the expansion was very big. These are the nucleotides that are usually trimmed when you map a read on the reference that correspond to the extra expansion that is present in these samples. And you can see clearly that this is a mosaic where there are many fragments of different length. But here, the number of repetitions that we could measure, it has been reaching a number that is close to 1,700. So we have an expansion that is up to 25 kb. And by looking even more in deep in other samples, we have been able to characterize the longest expansion that was a little bit shorter than 50 kb. So this is astonishingly to be able to characterize a single sequence that is almost 50 kb. And by combining also the technologies, it's possible at this point to do many things. And so I'm closing by telling you that, yes, sequencing is like fishing, but the technology that we have available to go fishing allow us to choose the right combination of hook, that is the target sequencing that we are using, and the bait, that is the sequencing technology that we are using, in order to properly characterize even very long fragments with a very accurate technology. So I'm at the end, I'm going to acknowledge my group, in particular our collaborators from University of Rome, and the person of my group who mainly contributed to this work, Marcia Rossato, Massimiliano Alfano, Valentina Grosso, Simone Maestri, and I thank you for your attention.
Thanks very much, Massimo, for this fantastic presentation. As a reminder, please submit your questions through the chat feature of the platform or raise your hand to ask your questions directly from our speakers. And now we will move on to our last speaker today, who is Dr. Edward Oakeley, Associate Director of Genomics at Novartis. Over to you, Edward.
Thank you very much. I would like to welcome you to something a little different. So first of all, I'm going to start with some background information. So genomes are very untidy, and really you can't exaggerate this enough. There are many variables within genomes, and really the concept of heterozygous genomes is really a very, let's say, antiquated respect for us. However, there are some challenges when we face the combination of both heterozygous and homozygous genomes simultaneously. In particular, when we're facing small short reads, we very often have the problem of phasing, and it can be very challenging to phase precisely how various bits of the genome rearrange, especially when we don't have a clear view of exactly what the genome is supposed to look like. However, there can be some challenges within genomes, in particular, as you know, we just heard, repeats can be very problematic. However, there are some exciting alternative options. Now, the first thing I would like to share with you is the idea of massively multiplexed sequence. So in the first case, what we can do is with the new Sequel 2 instrument, we can sequence up to 12,000 amplicons simultaneously using a combination of inline indexes together with smart belt indexes. So in this way, we can multiplex easily 1536 amplicons using row and column indexes, and then we can multiplex essentially quite a lot of multiplexes simultaneously using the smart belt indexes at the same time. However, okay, what am I saying? Okay, sequencing is all very well, but you know, it's not really a modern technology. Now, what if we could amplify with only one primer? Okay, let's have a little thought experiment. So what we can have is we can think about a PacBio sequencing, but instead of thinking about it at the level of a smart belt index, what about if we actually thought about using a primer that was directed against the genome? And in particular, what about if we thought about using a primer to amplify the PacBio library? Interesting. So let's have a little thought. Okay, the first thing we can do is we can make a size elf selection in order to make sure that we have a very tight size selection on our library. Then we can do a snowflake amplification. Now, a snowflake amplification is not quite like a rolling circle amplification because you can prime at both strands. So instead of amplifying in a linear manner, you actually amplify in an exponential manner or even more. Essentially, the longer the insert, the more rapidly you amplify. And you end up generating very quickly an absolutely enormous amount of product. Now, when you actually look at this, it suddenly ends up looking absolutely enormous. And this is because you have this rolling circle amplification. But when you then shear it again, in order to break it up into little pieces, the single-stranded bits of the genome shear much more easily than the double-stranded bits. So you end up getting it back more or less exactly to the original size that you had before, which is really cool. Because now, when you sequence it, ta-da, you end up with basically your gene of interest amplified as an absolutely amazing quantity. And you can also amplify this very, very easily. So essentially, if you target your gene clearly with a single primer, you can then amplify essentially to microgram quantities very, very easily. Now, what about RNA? Now, of course, as soon as you start thinking about isoseq, you can then start thinking about what if we could amplify a specific transcript instead. Now, of course, if you make double-stranded cDNA, you can then make a library as before. But now, if you prime with a primer at the the 5' end of the sequence, you can then make sure that you get a full-length transcript. Or if you have a gene fusion or some other thing, you could make sure that you have an amplification of that as well. And essentially, the amplification is again much the same. So, in summary, we can make a bunch of interesting applications. We can think about the understanding of complex regions. We can think about where is my gene and where is it complete. And we can think about repeat expansions. And then, of course, we can think about RNA-seq as well. Okay, thank you very much. And yeah, that's it.
Thank you very much, Ed, for this great presentation. Now, I would like to open the floor for the Q&A session. Please submit your questions through the chat feature of the platform, or raise your hand to ask a question directly from our speakers. And while we await for the first kind of questions to come in, I wanted to invite all our speakers, Patrick, Massimo, and Ed, to ask them if they wish to comment on anything that was addressed during the presentations. If there are any aspects that you would like to discuss, any points of your presentations.
Yeah, thank you very much, Esther. Let me first give my big thanks to Massimo and Ed for their very great presentations, which I think very nicely illustrate with clear and concrete examples how either long or short reads technology can be used in isolation or in combination, and also illustrates the specificities and the benefit and limitations of some of the technologies depending on the application. So really a big thanks to both of them. Having said that, maybe let me light the fire with the first question that was of course not prepared. So Ed, be ready. As always, I like very much the creativity in the way that you exploit your knowledge to identify new ways of working. Following the snowflake amplification, have you ever thought of putting your material directly on a nanopore sequencer and then generate a good consensus by simply aggregating the repeats of your fragments sequenced?
So I think the nanopore sequencer, unfortunately, is not a great deal for nanopore sequencing, because, for consensus sequencing, because it doesn't actually give you consensus. Or maybe I'm missing the point.
The point was that in your snowflake, you're basically generating a large tandem array of repeats because the initial molecule is circular. So basically, if you, instead of breaking it, if you put it in a nanopore, you will have a sequence that will give you the plus, the minus strand of your fragment of interest multiple times. And then using bioinformatics, you simply cut at the junction and reassemble all of it to generate a consensus. That was the idea behind.
Yeah, that's a fair comment. I think the advantage with the PacBio is just the better error rate. So when you have sort of, let's say, an 18 kb read, especially if it's a consensus sequence already, it amplifies very, very easily.
Absolutely. Yeah. Thank you. No, I don't know, Esther, I hand to you for any question coming from the participants.
Hi, Patrick. Yes, so we have received the following one. If we think forwards 10 years, is there any reason, cost, accuracy, other, that long read is not the obvious choice for the majority of applications in clinical or research applications?
So I can start to answer, and then I let, of course, Massimo and Ed complement. But you know, 10 years down the line, I like to compare this evolution with the small calculator that you have. If I mean, you're too young to know that, but when I remember when I was a child, when the calculator appeared, it was costing a lot, and we could do simple, very simple calculation. Now it costs nothing, and we can do very complex math. Clearly here, you know, of course, we know that the technology will evolve. Will we, you know, we are close to the $100 genome. We will be even going lower. I think that I would distinguish the needs or the interest of sequencing complete genome as opposed to very specific regions, because in clinical, both can be used, and I think that both will be necessary. So that's basically my answer to this question. But of course, I'm happy to have a Massimo or Ed complement.
So I think yeah, basically it's a good comment, but ultimately I think long read sequencing will win just because it's a good technology for phasing genomes, and ultimately when genomes become more heterozygous than homozygous, this is something which we will have to face.
Thank you very much. Massimo, would you like to comment on this question? Massimo, you are muted.
Oh, I could not reactivate my audio. Now it's okay. Okay, I can, yeah. For me, the point is always related to what you want to fish. We don't have a perfect technology. Short reads, very accurate. Long reads, not so accurate. If you work on a Mendelian disease, that's fine. You may use even PacBio and this CCS and so on. But if you have a cancer, if you have a matrix which is very complicated, using this kind of technology is still not the best solution. So I think at the moment we don't see the possibility of using a single technology to do everything. Maybe in 10 years time we will have another platform which will be able to go long and also to be accurate, but for the moment the current technologies do not allow to reach this point.
Are you sure?
Yes, I mean, for me, the best technology that I see on the horizon is quantum biosystems. That is a system that it's a nanopore that is measuring the tunneling current that should be so precise. But for the rest, no. Are you sure?
Yeah, I think the sequel to is Q60 to Q90, which I think is good enough.
Thank you very much. I would like to move on to the second question now. Can we apply CRISPR-Cas enrichment on viral pathogens such as SARS-2 in a clinical diagnostic setting?
I'm sorry, I did not understand the question.
Can you repeat the question, Esther, please?
Yeah, sure. Can we apply CRISPR-Cas enrichment on viral pathogens such as SARS-2 in a clinical diagnostic setting?
Oh, yeah, the answer is, of course, yes. The point is that in a clinical diagnostic setting, you need performance which are probably higher than the performance that this technology is offering at the moment. So in my opinion, it's a very promising technology, and it has to improve a little bit in terms of also of throughput before to become usable in a clinical setting. But in principle, yes, of course, it's already working.
Thank you very much. The next question is one that we received. This is for you. What do you mean when genomes become more heterozygous than homozygous?
So I think the genomes that we face in our individuals are more heterozygous than homozygous, but the traditional genome is more homozygous, and that is a problem because ultimately we face a lot of chimeras between genomes that have been assembled.
Thank you very much. The next question. Can we use long-read-seq for gene expression-seq and perhaps measure miRNAs?
I can start and then I'll let Ed or Massimo complete. Clearly, for classical RNA, especially when it's about quantifying isoforms, full-length isoforms, isoseq, for example, or on PacBio or full-length RNA sequencing on Oxford Nanopore can be used, although the quantification is not as accurate as it can be with shorter reads, although here we talk about partial sequences of the transcript. Now, when we talk about microRNAs, unless the protocol is using an amplification that generates concatenators of the sequence with the risk of introducing a bias in the quantification, personally, I don't really see the benefit of quantifying a 22 nucleotide RNA sequence with a technology that can generate megabases sequences.
Yeah, if I can say something, I fully agree with you, Patrick. There is no reason to use a long-read technology in order to sequence a short fragment. The only advantage is that if you use, for instance, Oxford Nanopore, you use a very cheap sequencer that allow every laboratory to do something like this. But now we have other sequencers that cost very little, that are probably more accurate than that for microRNA.
Yep. Maybe to complement, thank you, Massimo. For microRNAs, there are other type of technologies, either hybridization based or capture based, that are very interesting and that can be used to profile microRNAs. It really again depends on the question. The main question here being, do we want to profile known microRNAs for which we can have probes or capture or a system or even PCR, or do we want to discover new microRNAs where obviously sequencing is absolutely needed? So again, depending on the question, the different approach has to be selected.
Thank you very much. It seems like we don't have any more questions. Please, if you would still like to ask anything from our speakers, Ed, Massimo or Patrick today, please submit it now or raise your hand to ask a question directly from our speakers. Great. Well, it looks like we don't have any more questions. If there is still any, if you still have any questions unanswered, please feel free to email us and we would be very happy to feedback your question to our speakers. Patrick, Massimo and Edward will be speaking at our upcoming Next Genomics UK Congress, which is taking place this November in a virtual format. If you would like further information on the Congress or would like a copy of the conference programs, please contact the marketing department or visit the conference website. Now, I very much would like like to say a big thank you to all our speakers who put a lot of time and effort into making this workshop as interesting and valuable as possible. We very much enjoyed it. Thank you very much once again, Ed, Patrick, and Massimo. Thank you very much all for attending this workshop. We hope you enjoyed it and have a great rest of your day. And we look forward to welcoming you at one of our future events.
Well, thank you very much, Esther. Let me, in return, thank you for the organization. Everything went so smoothly, and that's great to see. Of course, also would like to thank very much my two friends, Massimo and Ed, for the great presentation. As always, a pleasure to interact with you in these difficult times. It's not possible to talk more about genomics with a more decent drink than a tea and a headset, but let's make sure that we continue this way. And I wish you all a good day.
Thank you very much. Thanks very much once again, everyone, and hope you have a lovely rest of your day.
Thank you.
Thanks very much. Bye.